New method compresses terabytes of genomic data into gigabytes. Genomic researchers used to be able to store their datasets on a laptop, but with so many whole genomes available now to study, the resulting big datasets must be stored in the cloud, resulting in more expensive, slower, and more unwieldy computations. A new method developed at Cornell provides tools and methodologies to compress hundreds of terabytes of genomic data into gigabytes, once again enabling researchers to store datasets in local computers. Their paper enabling efficient analysis of biobank-scale data with genotype representation graphs, published December 5th in Nature Computational Science. Even just a few years ago, the data we were studying usually wasn't whole genome sequencing data, which meant only a small fraction of the genomes were being measured rather than their entire genome, and because of that, the size of data wasn't so crazy, said April Wei, assistant professor of computational biology at the College of Arts and Science. Raw data size can now run into the petabyte, said co-author Drew DeHaas, computational genetics programmer in the College of Agriculture and Life Sciences. Wei had always wanted to develop methods to utilize biobank-scale data for doing research because of the richness of the information available, but many of the things she wanted to do were not possible because of the computational cost and challenge. This inspired her, she said, to tackle the compression problem, which led to the Genotype Representation Graph (GRG) method, which uses graphs to manage the data. Graph-based methods have long been used in computer science and other fields to provide a clear framework for solving challenging problems, DeHaas said, but prior to GRG had not been applied to a data compression solution in genomic at the biobank scale. Wei, trained as a population genetics, had deep familiarity with graphs used in population genetics, although GRG is designed quite differently. Unlike conventional matrix-based representations, GRG represents genotypes as a graph, where relationships between individuals are captured through shared mutations in their genomes. The GRG data structure not only encodes genotypic information more intuitively, compactly, but also facilitates efficient graph-based computations for advanced analysis, said co-author Vinkin Ban, doctoral student in the field of computational biology. GRG compresses the data while focusing on scalability and faithfully representing the data according to Wei. The great benefit of utilizing graphs for compression is that we can do computations with graphs without the need to decompress the data, she said. Also, specific algorithms could be developed to do things that people couldn't do with older formats. So there are potentially more benefits. Because the GRG enables researchers to analyze the same data more efficiently, it also lowers costs. The researchers' paper posted first on Biobank-Scale and Wei said GRG has already generated instant interest among other scientists who have begun testing the method on different data. The research was supported in part by a grant from the National Institute of Health. Thank you for watching.